To everyone's surprise, the debate has been resolved by DNA. 30,000-year-old DNA extracted in 1997 from the very bones that started it all in 1856. Having evolved in order to store information securely and transmit it from one generation to the next, DNA, no surprise, shows great chemical stability. It doesn't degrade spontaneously or react readily with other molecules, but it is not impervious to chemical damage. At the moment of death, the body's genetic materials, like all its other constituents, become susceptible to a horde of would-be degraders, reactive chemicals and enzymes that break down the molecular fabric. These chemical reactions require the presence of water, so DNA may be preserved if a corpse dehydrates fast enough. But even under ideal preservation conditions, the molecule is likely to survive perhaps 50,000 years at the absolute maximum. To obtain a legible DNA sequence from a 30,000-year-old Neanderthal remains preserved imperfectly was therefore a tall order at best. But Svante Paabo, a tall, laconic Swede at the University of Munich, decided to have a crack at the problem. If anyone could do it, he was the one. Paabo had pioneered work on the retrieval of so-called ancient DNA. He had scored sequences from Egyptian mummies, frozen mammoths, and the 5,000-year-old Iceman who melted out of an alpine glacier in 1991. Despite this impressive resume, though, the prospect of drilling into a precious Neanderthal relic to look for intact DNA, if indeed any was to be found inside, was daunting. As his archaeologist colleague Ralf Schmitz recalls, it was like getting permission to cut into the Mona Lisa. Matthias Krings, Paabo's graduate student, took on the project. He was pessimistic at first, but favorable early analyses to assess the bone's state of preservation emboldened Krings to press ahead. His search for viable DNA was focused not in the cell's nuclei, as one might expect, which are scattered throughout the cell outside the nucleus and produce the cell's energy. Each mitochondrion contains a small loop of DNA, some 16,600 base pairs in length. And because there are from 500 to 1,000 mitochondria in every cell, but only 2 copies of the genome proper in the nucleus, Krings knew that those decaying Neanderthal bones were much more likely to yield intact mitochondrial sequences than intact nuclear ones. Furthermore, since mitochondrial DNA, mtDNA, had long been a staple of studies of human evolution, Krings would have plenty of modern human sequences against which to make comparisons. A major worry for Krings and Paabo was contamination. In the past, a number of claimed successes at sequencing ancient DNA had proved to be erroneous when the sequence turned out to be from a modern source that had contaminated the sample. Every day, each of us sloughs off a vast number of dead skin cells, showering our DNA into the environment to wind up we know not where. The polymerase chain reaction, PCR, with which Krings expected to amplify the stretch of mtDNA he hoped to find, is so sensitive that it can act upon a single molecule, amplifying any DNA it might encounter, regardless of whether the source is ancient or still kicking. What if the Neanderthal DNA was too degraded for PCR to work? But the reaction proceeded nevertheless, amplifying a DNA sequence from an invisible contaminating particle that had flaked off Krings himself. Krings might then have to explain how he and the Neanderthal happened to have the same mtDNA sequence, a result unlikely to please the young man's boss and even less his parents. To ensure against this possibility, Krings and Paabo arranged for a separate laboratory, Mark Stonekings at Pennsylvania State University, to replicate the study. Contamination might occur there too, but probably not with DNA from Krings, a continent away. And if both labs obtained the same result from the sample, it would be reasonable to suppose they had found a bona fide Neanderthal sequence. "I can't describe how exciting it was," says Krings of the moment he first glimpsed the sequencing results. "Something started to crawl up my spine." Although, as feared, some sequences showed evidence of contamination, in others he could see something wondrous, a collection of intriguing similarities to, and differences from, the modern human sequence. Piecing together segments, he was able to reconstruct a Neanderthal mtDNA stretch running 379 base pairs. But the results weren't yet in from Penn State. Those sequences, however, proved to be the same. The identical 379 base pairs. That's when we opened the champagne, Krings recalls. The Neanderthal sequence had more in common with modern human mtDNA sequences than with those of chimpanzees, telling us that Neanderthals were unquestionably part of the human evolutionary lineage. At the same time, however, there were more dramatic differences between the Neanderthal sequences and all 986 available sequences of modern human mtDNA, to which Krings compared his sample. And even the most similar of those 986 sequences still differed from the Neanderthal one by at least 20 base pairs, or 5%. Subsequently, mtDNA has been sequenced from two other Neanderthals, one found in southwest Russia, the other in Croatia. The sequences, as expected, are not identical to the original one. We would expect to see variation among Neanderthal individuals, just as we would among modern humans. But they are similar. The sum of the genetic evidence leads us to conclude that while Neanderthals do have their place on the evolutionary tree of humans and their relatives, the Neanderthal branch is a long way from the modern human limb. If, when they encountered each other in Europe 30,000 years ago, Neanderthals and moderns had indeed interbred, Neanderthal mtDNA sequences would have entered the modern human gene pool. That we see no evidence of such Neanderthal input implies that modern humans eliminated the Neanderthals rather than interbreeding with them. But whether they achieved the lethal result by direct confrontation or by more subtle means is something the